Hello YouTube, my name is Neil and we're studying for the MCATs today. We're in the Biochemistry review. Chapter 6, DNA and Biotechnology. How do all the cells of our body know what job to do? The cells that make up the human body have nucleic acids that instruct the cell on how to function. The nucleic acid, deoxyribonucleic acid, DNA, stores the information in our cells and selectively shares that information when appropriate. DNA is a molecule that can be passed from generation to generation. DNA can be replicated in a carefully regulated process designed to keep the genome safe from degradation and free from errors. Seemingly small changes in the genetic code can result in life-threatening and even life-incompatible alterations to protein structure and function. In this chapter, the unique structure of DNA will be discussed along with replication and repair processes. The primary focus will be on eukaryotic processes, but there will be some review of prokaryotic genetics to help us better understand the molecular basis of life. Much of the advancement of medicine in the past 2 decades has been due to our increased understanding of molecular genetics, which has led to the creation of an entire biotechnology industry centered around genomics and the utilization of nucleic acids for various diagnostic tests and therapeutic interventions. We will also take a look at some of these important principles in this chapter. 6.1 DNA structure. There are 2 chemically distinct forms of nucleic acids within eukaryotic cells: deoxyribonucleic acid, DNA, and ribonucleic acid, RNA, are polymers, each with distinct roles that together create the molecules integral to life in all living organisms. DNA is the focus of this chapter, and RNA will be discussed in more detail in Chapter 7. The bulk of DNA is found in chromosomes in the nucleus of eukaryotic cells, although some is also present in mitochondria and chloroplasts. Let's write down deoxyribonucleic acid and ribonucleic acid are polymers. Nucleosides and nucleotides. DNA is a macromolecule and it is essential to understand how this molecule is constructed. DNA is a polydeoxyribonucleotide that is composed of many monodeoxyribonucleotides linked together. The nomenclature for nucleic acids can be complicated, so the terms have been defined here. Nucleosides are composed of a 5-carbon sugar pentose bonded to a nitrogenous base and are formed by covalently linking the base to C1' of the sugar, as shown in figure 6.1. Note that the carbon atoms in the sugar are labeled with a prime symbol to distinguish them from the carbon atoms in the nitrogen base. All right. Nucleoside. Examples of nucleosides: adenosine deoxythymidine. And what we have is a sugar bonded to a nitrogen base, which is a pretty complicated nitrogen base. It's a 6-ring and a 5-ring. What do we call those? All right, so nucleotides, on the other hand, are formed when one or more phosphate groups are attached to C5' of a nucleoside Often these molecules are named according to the number of phosphates present. Adenosine di- and triphosphate, ADP and ATP, for example, gain their name from the number of phosphate groups attached to the nucleoside adenosine. These are high-energy compounds because of the energy associated with the repulsion between closely associated negative charges on the phosphate groups, as shown in Figure 6.2. Nucleotides are the building blocks of DNA. So a nucleoside with ADP or ATP attached to it is what a nucleotide is. Nucleoside plus ADP, ATP. Did you have a adenosine monophosphate? You don't see that, do you? Probably could, though. GTP. AMP, there you go. Adenosine monophosphate. AMP, ADP, ATP. And let's write adenosine, nitrogen base, got some nitrogens all around our rings. We have double bonds. And then this nitrogen is linked to a sugar. They have this bond extending. Oh no, because there's another carbon on the end of that, probably. Or maybe a hydrogen. Because that's probably a hydrogen, not a carbon, right? Or maybe it is a carbon. This is a substituted sugar. And then a CH2OH over here, our last carbon. 5-carbon sugar. Furanose. That looks good. Should we number these carbons? 1 through 5. Carbon number 5. We got 1 over here. We got 6 over here. Got 9 over here. Should we just fill them all in? Because this is kind of confusing. Got 4 in here. We got 3 is the nitrogen. One, 2, 3, 4. So there's got to be 5 over here. 6, 7, 8, 9. 1, 2, 3, 4, 5. Oxygen doesn't get a number. Ha! Alright. So that looks pretty complicated, but that's adenosine. Then you got deoxythymidine, but I'm not going to draw that. I'm just going to draw adenosine for now. Triphosphate, you have the phosphate attached to this CH2. Looks like it's probably an addition reaction. You probably lose water in the process and attach 1, 2, or 3 phosphates. So let's draw that in as well. If you could kick off water, then you can add oxygen and a phosphate. And you can add more. That is adenosine for you. Nucleic acids are classified according to the pentose they contain, as shown in figure 6.3. If the pentose is ribose, the nucleic acid is RNA. If the pentose is deoxyribose, ribose with 2 OH groups replaced by hydrogens, then it is DNA. Okay, so these are not those are not carbons, those are hydrogens. So ribose and replacing oxygens with hydrogens makes deoxyribose. So adenosine is DNA, right? So let's just, in another color, if we took off this, then you'd have deoxy. Nucleoside. This is a nucleoside, and we can make a deoxy nucleoside out of it. Is this ribose in this case? It is ribose. No. This is not. Yeah, this is. This is a 5-carbon sugar. So why doesn't it have the extra carbon up here? Maybe it does. This is a carbon, and then OH. So this carbon and then OH. All right. The nomenclature for the common bases, nucleosides and nucleotides, is shown in table 6.1. Note that there is no thymidine listed, only deoxythymidine, because thymidine appears only exclusively in DNA. thymidine. There's a deoxythymidine. So for each of them, there's adenosine, then deoxyadenosine, and the nucleotides they make are AMP for adenosine, GMP for guanine, CMP for cysteine, or cytosine rather, UMP for uracil, and DTMP, so I guess di is the smallest you can go. Oh no, it's monophosphate, but it's deoxythiamine is the only option. There's no RNA version because it's only in DNA form. Should we just list adenine, guanine, cytosine, uracil, thymine. Adenine makes adenosine. Guanine makes guanosine. Cytidine. Uridine. And thymidine. I should write deoxy. Sugar phosphate backbone. The backbone of DNA is composed of altering sugar and phosphate groups. It determines the directionality of the DNA and is always read from 5 to 3. It is formed as nucleotides are joined by 3' to 5' phosphodiester bonds. That is, a phosphate group links the 3' carbon of one sugar to the 5' phosphate group of the next incoming sugar in the chain. Phosphates carry a negative charge, thus DNA and RNA strands have an overall negative charge. DNA is negative. So here's the 5 carbon, here's the 3 carbon, so this end is the 5, this end is the 3, and a 3 to 5 linkage is the 3 to 5 phosphodiester bond. And these are the sugars, and then you have the base coming off the top here. So to make DNA to this same adenosine molecule, you would link the 3 to the 5 of the next incoming with a phosphate. So it's like this phosphate would be between 5 and 3 of different molecules. So should I put it over here? Yeah, because it's literally like coming off here. It would attach to another molecule's 3 right there. All right, each strand of DNA has distinct 5' and 3' ends, creating a polarity within the backbone, as shown in figure 6.4. The 5' end of DNA, for instance, will have a hydroxyl or phosphate group bonded to the C5 sugar, or the C5, 5 carbon of the sugar, while the 3 end has a free hydroxyl group on C3 of the sugar. So it starts with the 5 end. We'll have an OH or a phosphate group, while the 3 end has a free hydroxyl. The base sequence of a nucleic acid strand is both written and read in the 5' to 3' direction. Thus, the DNA strand in figure 6.4 must be written 5' ATG 3', or simply ATG. DNA sequences can also be written in slightly different ways. If written backwards, the end must be labeled 3' GTA 5'. The position of phosphates may be shown, so PA, PT, PG. D may be used as shorthand for deoxyribose, so DA, DT, DG for DNA. DNA is generally double-stranded, DS DNA, and RNA is generally single-stranded, SS RNA. Exceptions to this rule may be seen, especially in viruses, or described in Chapter 1 of Biology. Purines and pyrimidines. Purines and pyrimidines. Adenosine, guanine, cytosine, uracil, thymine. Purines are adenine and guanine. Pyrimidines are cytosine, uracil, and thymine. There are 2 families of nitrogen-containing bases found in nucleotides, purines and pyrimidines. The bases described below and shown in figure 6.5 represent the common bases in eukaryotes. However, it should be noted that exceptions may be seen in tRNA and in some prokaryotes and viruses. Purines contain 2 rings in their structure. The 2 purines found in nucleic acids are adenine and guanine. Both are found in DNA and RNA. Pyrimidines contain only 1 ring in their structure. The 3 pyrimidines are cytosine, thymine, and uracil. While cytosine is found in both DNA and RNA, thymine is only found in DNA, and uracil is only found in RNA. Thymine, DNA, uracil, RNA. And the difference between them is 1 carbon, a methyl group. on the one carbon. Oh no, this is not the one carbon. This would be like the... I'm not really sure. I think this is probably... Actually, yeah, this is got the double bond. This has got the substitution on it. But this is nitrogen. Nitrogen's a better substitution, right? We start with this is the 1. So this will be the 5. 1, 2, 3, 4, 5. 5 carbon. Should I say that this is the one that gets attacked? This is the electrophile because of the leaving group. This is the neutrophile, electrophile. I kind of want to draw these out a little bit. Is there a way to copy this? Should be a way to copy it, right? Whoops. Maybe not. I bet I could do it with my keyboard, right? Just gonna make a bunch of these. Got adenine, guanine, and the pyrimidines. cytosine, uracil, and thymine. Bases commonly found in nucleic acids. All right, purines and pyrimidines are examples of biological aromatic heterocycles. In chemistry, the term aromatic describes any unusually stable ring system that adheres to the following four specific rules. The compound is cyclic, the compound is planar, the compound is conjugated with alternating single and multiple bonds or lone pairs, creating at least one unhybridized p orbital for each atom in the ring. The compound has 4n plus 2, where n is any integer, pi electrons. That's how many pi electrons there is. This is called Huckel's rule. N is any integer. 4n plus 2 is how many pi electrons there are. This is called Huckel's rule. Let's write down Huckel's rule. 4n plus 2 pi electrons. The most common example of an aromatic compound is benzene. Many different structures obey these rules. In an aromatic compound, the extra stability is due to the delocalized pi electrons, which can travel throughout the entire compound using available molecular orbitals. All 6 of the carbon atoms in benzene are sp2 hybridized, and each of the 6 orbital overlaps equally with its 2 neighbors. As a result, the delocalized electrons form 2 pi electron clouds, one above and below the plane of the ring, as shown in figure 6.6. This delocalization is characteristic of all aromatic molecules, and because of this, aromatic molecules are fairly unreactive. Heterocycles are ring structures that contain at least 2 different elements in the ring. As shown in figure 6.5 earlier, both purines and pyrimidines contain nitrogen in their aromatic rings. Nucleic acids are thus imbued with exceptional stability. This helps to explain the utility of nucleotides as the molecule for storing genetic information. Watson-Crick model. Putting this information together, we can start looking at the Watson-Crick model of DNA structure. In 1953, James Watson and Francis Crick presented one of the landmark findings of modern biology and medicine, the 3-dimensional structure of DNA. They were able to deduce the double helical structure, or double helical nature of DNA, and propose specific base pairing that would be the basis of a copying mechanism. In the double helix, 2 linear polynucleotide chains of DNA are wound together in a spiral orientation along a common axis. The key features of the model, some of which have already been mentioned, are the 2 strands of DNA are anti-parallel, that is, the strands are oriented in opposite directions. When one strand has polarity 5' to 3' down the page, the other strand has 5' to 3' polarity up the page. The sugar phosphate backbone is on the outside of the helix with the nitrogen bases on the inside. There are specific base pairing rules, often referred to as complementary base pairing. As shown in figure 6.7, An adenine is always base paired with a thymine via 2 hydrogen bonds, and a guanine always pairs with cytosine via 3 hydrogen bonds. The 3 hydrogen bonds make the GC base pair interaction stronger. These hydrogen bonds and the hydrophobic interactions between bases provide stability to the double helix structure. Thus, the base sequence on one strand defines the base sequence on the other strand. Because of the specific base pairing, the amount of A equals the amount of T, and the amount of G equals the amount of C. Thus, total purines will be equal and total pyrimidines overall. These properties are known as Chargaff's rules. Watson, Crick, model. Anti-parallel. One goes this way, the other goes this way. 5' to 3', 5' to 3'. Let's draw these all on here. And what I'm going to do this. So we got the bases on the inside. So we got 5' to 3'. We got between A and T, we have two hydrogen bonds, and between G and C, we have 3 hydrogen bonds. So this one's stronger. You have hydrophobic interactions along with those hydrogen bonds. And you have, so it's not 3 hydrogens linked in a row like this. It's 3 to different carbons. Or nitrogens, I guess. So let's redraw this. And we have Chargaff's rules. A equals T, G equals C, A equals T, G equals C. So purines and pyrimidines are equal. Therefore, double helix of most DNA is a right-handed helix, forming what is called B-DNA, as shown in figure 6.8. The helix in B-DNA makes a turn every 3.4 nanometers and contains about 10 bases within that span. Major and minor grooves can be identified between the interlocking strands and are often the site of protein binding. Another form of DNA is called Z-DNA for its zigzag appearance. It is a left-handed helix that has a turn every 4.6 nanometers and contains 12 bases within each turn. A high GC content or a high salt concentration may contribute to the formation of this form of DNA. No biological activity has been attributed to Z-DNA, partly because it is unstable and difficult to research. So science made fake left-handed DNA in the lab, and it doesn't really work, apparently. But we have B-DNA, which is right-handed. Denaturation, and reannealing. During processes such as replication and transcription, it is necessary to gain access to the DNA. The double helical structure or nature of DNA can be denatured by conditions that disrupt hydrogen bonding and base pairing, resulting in the melting of the double helix into two single strands that have separated from each other. Notably, none of the covalent links between the nucleotides and the backbone of the DNA break during this process. Heat, alkaline pH, and chemicals like formaldehyde and urea are commonly used to denature DNA. Denatured single-stranded DNA can be reannealed, brought back together, if the denaturing condition is slowly removed. If a solution of heat denatured DNA is slowly cooked, for example, then the two complementary strands can become paired again, as shown in figure 6.9. If a solution of heat denatured DNA is slowly cooled, for example, then the two complementary strands can become paired again. Double-stranded DNA, denatured or straight, double-stranded again. Such annealing of complementary DNA strands is also an important step in many laboratory processes, such as polymerase chain reactions, PCR, and in the detection of specific DNA sequences. In these techniques, a well-characterized probe DNA with known sequence, is added to a mixture of target DNA sequences. When probe DNA binds to target DNA sequences, this may provide evidence of the presence of a gene of interest. This binding process is called hybridization and is described in further detail later in this chapter. What is the difference between a nucleoside and a nucleotide? The nucleotide has a phosphate attached to the 5-carbon of the sugar of a nucleoside. 2. What are the base pairing rules according to the Watson-Crick model? Base pairing rules are G to C and A to T. In RNA, A to U. Are there more base pairing rules? They run anti-parallel. Sugar phosphate backbone is on the outside. And equal number of AT and GC. What are the three major Major structural differences between DNA and RNA. DNA is deoxyribonucleic acid. RNA is not specifically the 2 carbon on the sugar is deoxygenated. That makes for well, all right, so thymine is deoxygenated. Uracil, since it's an RNA, shouldn't it be oxygenated? Other structural differences between RNA and DNA. DNA is double helix, RNA is not. Other differences. I guess the difference between the deoxygenated 2 carbon, the difference between uracil and thymine. How does the aromaticity of purines and pyrimidines underscore their genetic function? They're very stable, so they're good for storing information like DNA. They won't get corrupted easily. If a strand of RNA contained 15 cytosine, 15 adenine, 35 guanine, and 35 uracil, would this violate Chargaff's rules? Why or why not? Cytosine, adenine, guanine, uracil. RNA, so no thymine, it's uracil. G equals C, and A equals T or U, so it would break the rules. Let's check the answers. (6.1) Number 1, nucleosides contain a 5-carbon sugar pentose and nitrogen base. Nucleotides are composed of a nucleoside plus 1 to 3 phosphate groups. Number 2, A pairs with T in DNA, or U in RNA using 2 hydrogen bonds. C pairs with G using 3 hydrogen bonds. Number 3, DNA contains deoxyribose while RNA contains ribose. DNA contains thymine while RNA contains uracil. Usually DNA is double-stranded while RNA is single-stranded. There you go. Number 4, the aromaticity of nucleic acids makes these compounds very stable and unreactive. Stability is important for storing genetic information and avoiding spontaneous mutations. Number 5, this does not violate Chargaff's rules. RNA is single-stranded, and thus the complementarity seen in DNA does not hold true for single-stranded RNA. C does not necessarily equal G. A does not necessarily equal U. Oh, there you have it. For RNA, Chargaff's rule does not hold. That makes sense, because I guess this property comes out of the fact that G will only bond to T, or C, and A will only bond to T. So that doesn't have to hold for RNA. Eukaryotic chromosome organization. There are over 6 billion bases of DNA in each human cell. It is important for the cell to organize these bases effectively. These bases must be replicated during the cell cycle and also utilized in gene expression for normal cellular functions. In humans, DNA is divided up among the 46 chromosomes found in the nucleus of the cell. The supercoiling of the DNA double helix does not provide some compaction, but much more is necessary. The supercoiling of the DNA double helix does provide some compaction, but much more is necessary. 46 chromosomes, 2 copies of 23. Histones. Let's write down chromosomes, histones. The DNA that makes up a chromosome is wound around a group of small basic proteins called histones, forming chromatin. Chromatin. There are 5 histone proteins found in eukaryotic cells. 2 copies each of the histone proteins H2A, H2B, H3, and H4 form a histone core, and about 200 base pairs of DNA are wrapped around this protein complex, forming a nucleosome, as shown in figure 6.10. Under an electron microscope, the nucleosomes look like beads on a string. The last histone, H1, seals off the DNA as it enters and leaves the nucleosome, adding stability to the structure. Together, the nucleosomes create a much more organized and compacted DNA. So H2A, H2B, H3, and H4 in the nucleosome. In the core, and H1 sealing off on the outside. H2A, H2B, H3, H4 make the core or the nucleosome. I guess the nucleosome is DNA wrapped around the core. Plus DNA makes a nucleosome, and another H1 Histones are one example of nucleoproteins, proteins that associate with DNA. Most other nucleoproteins are acid-soluble and tend to simulate processes such as transcription. Histones are nucleoproteins. Heterochromatin and Euchromatin. DNA. So the core, I want to rewrite this, that the core plus All right. H2A, H2, the core plus DNA plus H1 makes a nucleosome. And histones are nucleoproteins, heterochromatin and euchromatin. The chromosomes have a diffuse configuration during interphase of the cell cycle. The cell will undergo DNA replication during the S phase of interphase, and having the DNA uncondensed and accessible makes the process more efficient. A small percentage of the chromatin remains compacted during interphase and is referred to as heterochromatin. So we're talking about S phase of interphase. Heterochromatin stays compact. During interphase. S. Heterochromatin appears dark under light microscopy and is transcriptionally silent. Heterochromatin often consists of DNA with highly repetitive sequences. In contrast, the dispersed chromatin is called euchromatin, which appears light under light microscopy. Euchromatin contains genetically active DNA. Both heterochromatin and euchromatin can be seen in the nucleus in figure 6.11. Euchromatin is genetically active. Telomeres and centromeres. As described later in this chapter, DNA replication cannot extend all the way to the end of a chromosome. This will result in losing sequences and information with each round of replication. The solution for our cells is a simple repeating unit TTAGGG at the end of the DNA, forming a telomere. Some of the sequence is lost in each round of replication, and it can be replaced by the enzyme telomerase. Telomerase is more highly expressed in rapidly dividing cells. Animal studies indicate that there are a set number of replications possible, and that the progressive shortening of telomeres contributes to aging. Telomeres also serve a second function. Their high GC content creates exceptionally strong strand attractions at the end of chromosomes to prevent unraveling. Think of telomeres as knotting off the end of the chromosome. So the telomere is TTAGGG. Telomerase centromeres, as their name suggests, are a region of DNA found in the center of the chromosome. They are often referred to as sites of constriction because they form noticeable indentations. This part of the chromosome is composed of heterochromatin, which is in turn composed of tandem repeat sequences that also contain high GC content. During cell division, the two sister chromatids can therefore remain connected at the centromere until microtubules separate the chromatids during anaphase. So, GC was the stronger of our two bonds. Yeah, GC is the 3 hydrogen bond flavor. Before you move on, assess your understanding of the material with these questions. What are the 5 histone proteins in eukaryotic cells? Which one is not part of the histone core around which DNA wraps to form chromatin? Well, H2A, H2B, H3, and H4 are in the core. H1 is not. Compare and contrast heterochromatin and euchromatin based on the following characteristics. Density of chromatin packing. Heterochromatin is denser than euchromatin. Appearance under light microscopy. Heterochromatin is darker than euchromatin. Transcriptional activity. Heterochromatin is inactive, while euchromatin is active. What property of telomeres and centromeres allows them to stay tightly raveled even when the rest of the DNA is uncondensed? I guess the high GC content. Those 3 hydrogen bonds make a difference. 6.3, DNA replication. Should we check the answers? Let's check the answers, I guess. Felt pretty confident about that. 6.2, histones H1, 2A, 2B, 3 and 8, and 4. 1 is not in the core. Chromatin is dense. We got dense, not dense, dark, light, silent, active. The high GC content increases hydrogen bonding, which makes telomeres and centromeres strong. Great. DNA replication. The DNA is an organism's blueprint that provides not only the ability to sustain activities of life, but also insight into our evolutionary past. The process of DNA replication is highly regulated to ensure as close to 100% perfection when making a copy of our genome as possible. DNA replication is necessary for the reproduction of a species and for any dividing cell. Strand separation and origins of replication. Origins. The human genome has about 3 billion base pairs packed into multiple chromosomes. The replisome, or replication complex, is a set of specialized proteins that assist the DNA polymerases. To begin the process of replication, DNA unwinds at points called origins of replication, or origins of replication. The generation of new DNA proceeds in both directions, creating replication forks on both sides of the origin, as shown in figure 6.12. Let's write down the replisome. DNA replication in both directions. The bacterial chromosome is a closed double-stranded circular DNA molecule with a single origin of replication. Thus, there are 2 replication forks that move away from each other in opposite directions around the circle. The 2 replication forks eventually meet, resulting in the production of 2 identical circular molecules of DNA. Eukaryotic replication must copy many more bases compared to prokaryotes and is a slower process. In order to duplicate all the chromosomes efficiently, each eukaryotic chromosome contains 1 linear molecule of double-stranded DNA having multiple origins of replication. As the replication forks move toward each other and sister chromatids are created, the chromatids will remain connected at the centromere. The difference between prokaryotic and eukaryotic replication patterns are shown in figure 6.13. Multiple origins of replication. And a centromere, linking the two linear strands. So let's write down in eukaryotes, you have multiple origins of replication. Helicase. Do we need to write centromere? Sister chromatids. Sister chromatids connected at centromere. There's a little bit of review, I guess. Helicase. Helicase is the enzyme responsible for unwinding the DNA, generating two single-stranded template strands ahead of the polymerase. Once open, the unpaired strands of DNA are very sticky in a molecular sense. The free purines and pyrimidines seek out other molecules with which to hydrogen bond. Proteins are therefore required to hold the strands apart. Single-stranded DNA binding proteins will bind to the unraveled strand, preventing both the reassociation of the DNA strands and the degradation of DNA by nucleases. As the helicase unwinds the DNA, it will cause positive supercoiling that strains the DNA helix. Supercoiling is a wrapping of DNA on itself as its helical structure is pushed even further toward the telomeres during replication. Picture an old-fashioned telephone cord that has been tangled on itself. To alleviate this torsional stress and reduce the risk of strand breakage, DNA topoisomerases introduce negative supercoils. They do so by working ahead of helicase, nicking one or both strands, allowing relaxation of the torsional pressure, and then resealing the cut strands. So a helicase unwinds DNA, single-stranded DNA binding proteins hold it in place. Hold ssDNA in place. Let's make these lowercase s as well. Supercoiling is a problem, so DNA topoisomerases nicking one or both strands, meaning cutting. Cut DNA, ssDNA, to reduce supercoiling. During replication, these parental strands will serve as templates for the generation of new daughter strands. The replication process is termed semi-conservative because one parental strand is retained in each of the two resulting identical double-stranded DNA molecules, as shown in figure 6.14. Each daughter strand gets half of the parent strand. Synthesis of daughter strands. So should we write down that it's semi-conservative? Meaning that if you start with this red guy, each daughter strand will get half of it. Synthesis of daughter strands. DNA polymerases are responsible for reading the DNA template or parental strand and synthesizing the new daughter strand. The DNA polymerase can read the template strand in a 3' to 5' direction while synthesizing the complementary strand in the 5' to 3' direction. This will result in a new double helix of DNA that has the required anti-parallel orientation. Due to this directionality of the DNA polymerase, certain constraints arise. Remember that the two separated parental strands of the helix are also anti-parallel to each other. Thus, at each replication fork, one strand is oriented in the correct direction for DNA polymerase, the other strand is anti-parallel. So let's write that DNA polymerase reads 3 to 5. Synthesizes 5 to 3. The leading strand in each replication fork is the strand that is copied in a continuous fashion in the same direction as the advancing replication fork. This parental strand will be read 3 to 5, and its complement will be synthesized 5 to 3. So that's a leading strand. The lagging strand is a strand that is copied in a direction opposite the direction of the replication fork. On this side of the replication fork, the parental strand has 5' to 3' polarity. DNA polymerase cannot simply read and synthesize on this strand. How does it solve this problem? Because DNA polymerase can only synthesize in the 5 to 3 direction. From a 3 to 5 template, small strands called Okazaki fragments are produced. As the replication fork continues to move forward, it clears additional space that DNA polymerase must fill in. Each time DNA polymerase completes an Okazaki fragment, it turns around to find another gap that needs to be filled in. So, leading strand, all good. Lagging strand is filled in with Okazaki fragments. Great. This process of DNA replication is shown in figure 615. The first step in the replication of DNA is actually to lay down an RNA primer. DNA cannot be synthesized de novo, that is, it needs another molecule to hook on to. RNA, on the other hand, can be directly paired with the parent strand. Thus, Primase synthesizes a short primer, roughly 10 nucleotides, in the 5 to 3 direction to start replication on each strand. These short RNA sequences are constantly being added to the lagging strand because each Okazaki fragment must start with a new primer. In contrast, the leading strand requires only one, in theory. In reality, there are usually a few primers on the leading strand. DNA polymerase 3 in prokaryotes and DNA polymerases alpha, delta, and epsilon in eukaryotes will then begin synthesizing the daughter strand of DNA in the 5-3 manner. The incoming nucleotides are 5' deoxy nucleotide triphosphates, DATP, DCTP, DGTP, and DTTP. As the new phosphodiester bond is made, a free pyrophosphate is released. So let's write down primase makes the 10 nucleotide primer. We don't need to know that it's 10, or we'll remember, but it's an RNA primer. And then DNA polymerase alpha, delta, and epsilon in humans will synthesize using deoxy nucleotide triphosphates. The RNA must eventually be removed to maintain sanctity of the genome. This is accomplished by the enzyme DNA polymerase I in prokaryotes or RNase H in eukaryotes. Then DNA polymerase I or DNA polymerase delta in eukaryotes adds DNA nucleotides where the RNA primer had been. DNA ligase seals the end of the DNA molecules together, creating one continuous strand of DNA. So RNase H removes primer, and DNA polymerase delta comes in and replaces RNA with DNA. And finally, DNA ligase seals ends. While prokaryotic DNA synthesis has been worked out in great detail and eukaryotic synthesis is considered to be very similar, there are a few differences in the enzymes involved. There are 5 classic DNA polymerases in eukaryotic cells, which are designated with the Greek letters alpha, beta, gamma, delta, and epsilon. Further research has revealed more polymerases named zeta through mu, but these are outside the scope of the MCAT. Table 6.2 highlights differences in the names of the enzymes associated with DNA replication in prokaryotic versus eukaryotic cells. Special attention should be paid to the eukaryotic DNA polymerases. DNA polymerases alpha, delta, and epsilon work together to synthesize both the leading and lagging strands. DNA polymerase delta also fills in the gaps left behind when RNA primers are removed. DNA polymerase gamma replicates mitochondrial DNA. Let's write this down. DNA gamma for mitochondria. DNA polymerase beta and epsilon are important in the process of DNA repair. DNA polymerases delta and epsilon are assisted by the pCNA protein, which assembles into a trimer to form the sliding clamp. The clamp helps to strengthen the interaction between these DNA polymerases and the template strand. So let's write DNA polymerase beta and epsilon are important in repair. I forgot to put the word polymerase in here. I don't like the way this gamma looks. Great. DNA polymerase D, delta, and epsilon are assisted by pCNA protein. A trimer to form the sliding clamp. And a chart with steps in replication and the differences in prokaryotic and eukaryotic cells. Origin of replication, one per chromosome, multiple in eukaryotic. Unwinding of DNA, helicase in both cases. Stabilization, single-stranded binding protein in both cases. Synthesis is synthesis of primers is primase in both cases. Synthesis of DNA is different. It's going to be polymerase 3 in prokaryotes, alpha, delta, and epsilon in eukaryotes. Removal of primers is going to be polymerase 1 in prokaryotes, RNAs in eukaryotes. Replacement of RNA with DNA is going to be polymerase 1 also in prokaryotes, DNA polymerase delta in eukaryotes. Joining of Ozaki fragments, DNA ligase in both cases. Removal of positive supercoils ahead of advancing replication forks. Topoisomerases in both cases. Also, there's a gyrase over here in the prokaryotic cell. Synthesis of telomeres, not applicable in prokaryotes. It's done by telomerase in eukaryotes. Did I ever write down telomerase? I did. Pretty sure, yeah, telomerase. Repair telomeres, make telomeres, whatever, telomeres. So Ozaki fragments. Do prokaryotes still have Ozaki fragments? I guess it still does, huh? All right. Replicating the ends of chromosomes. While DNA polymerase does an excellent job of synthesizing DNA, it unfortunately cannot complete synthesis of the 5' end of the strand. Thus, each time DNA synthesis is carried out, the chromosome becomes a little shorter. To lengthen the time that cells can replicate and synthesize DNA before necessary genes are damaged, chromosomes contain telomeres. As described earlier, telomeres are located at the very tips of the chromosome and consist of repetitive sequences with a high GC content. This repetition means that telomeres can be slightly degraded between replication cycles without loss of function. For each of the enzymes listed below, list the function of the enzyme and if it is found in prokaryotes, eukaryotes, or both. Helicase, both. Unwinds DNA. ssDNA binding protein. Both. Holds DNA in place. Primase. What makes the primer in both? Primase in both. DNA polymerase 3, that's a prokaryotic protein. And I guess it does the same as the alpha protein, as well as delta and epsilon. So it replicates, or synthesis of DNA. DNA polymerase alpha is the eukaryotic version that synthesizes. DNA polymerase 1 was that repairing, removal of RNA primers, and that's only in prokaryotes. RNase is the eukaryotic version, and it removes primers. DNA ligase seals the ends of DNA. I guess it works in both. DNA topoisomerase stabilizes DNA by nicking it, I guess. And it's in both prokaryotes and eukaryotes. Between the leading strand and lagging strand, which is more prone to mutations and why? I guess I'd say the lagging strand. Because for no reason. I mean, the leading strand is also not just one replication site. But there's going to be multiple replication sites, and they're going to have to seal in between them with another DNA polymerase like epsilon probably, repair. But I imagine that the Ozaki fragments on the lagging strand would be a place for mistakes. What is the function of a telomere? It seals up the end of the DNA strand, and it's repeated, so it's okay if it degrades a little bit because it's hard for DNA polymerase to replicate that part of the DNA because it's meant to stay coiled and tight and seal the DNA. Let's check those answers. Six, 3. The chart, pretty confident about. The lagging strand is prone to mutations because it must constantly start and stop the process of DNA replication. Additionally, it contains many more RNA primers, all of which must be removed and filled in with DNA. All right. Number 3, telomeres are the ends of eukaryotic chromosomes and contain repetitive sequences of non-coding DNA. These protect the chromosome from losing important genes from the incomplete replication of the 5' end of the DNA strand. Great. DNA repair. All right. DNA repair. Structure of DNA can be damaged in a number of ways, such as exposure to chemicals or radiation. DNA is very susceptible to damage, and if the damage is not corrected, it will be subsequently copied and passed on to daughter cells. Damage can include breaking of the DNA backbone, structural or spontaneous alterations of bases, or incorporation of incorrect base during replication. Any defect in the genetic code can cause an increased risk of cancer, so the cell has multiple processes in place to catch and correct genetic errors. This helps maintain the integrity and stability of the genome from cell to cell and from generation to generation. Oncogenes and tumor suppressor genes. Certain genes, when mutated, can lead to cancer. Cancer cells proliferate excessively because they are able to divide without stimulation from other cells and are no longer subject to the normal controls on cell proliferation. By definition, cancer cells are able to migrate by local invasion or metastasis, a migration to distant tissues by the bloodstream or lymphatic system. Over time, cancer cells tend to accumulate mutations. Mutated genes that cause cancer are termed oncogenes. Oncogenes primarily encode cell cycle-related proteins. Before these genes are mutated, they are often referred to as proto-oncogenes. Proto-oncogenes lead to oncogenes, which lead to cancer. The first gene in this category to be discovered was SRC, named after sarcoma, a category of connective tissue cancer. The abnormal alleles encode proteins that are more active than normal proteins, promoting rapid cell cycle advancement. Typically, a mutation in only one copy is sufficient to promote tumor growth and is therefore considered dominant. SRC causes sarcoma. Connective tissue cancer. And it's dominant because only one copy is sufficient. Tumor suppressor genes like P53 or RB, retinoblastoma, encode proteins that inhibit the cell cycle or participate in DNA repair processes. They normally function to stop tumor progression and are sometimes called anti-oncogenes. Tumor suppressor include P53 and RB. Anti-oncogenes. Mutations of these genes result in the loss of tumor suppression activity and therefore promote cancer. Inactivation of both alleles is necessary for the loss of function because in most cases, even one copy of the normal protein can function to inhibit tumor function. In this example, multiple mutations or hits are required. So I guess it's also dominant. So in this case, a hit causes you to get it. In this case, a hit doesn't cause you to get it because it itself is a tumor suppressor. So deactivating it is what causes you to get cancer. Proofreading and mismatch repair. Dominant requires 2 hits to knock out. Great. Proofread and mismatch repair. DNA polymerase moves along a single strand of DNA, building the complementary strand as it goes. When DNA polymerase is about almost 100% accurate, it does occasionally make errors. Proofreading. During synthesis, the 2 double-stranded DNA molecules will pass through a part of the DNA polymerase enzyme for proofreading. When the complementary strands have incorrectly paired bases, the hydrogen bonds between these strands can be unstable, and this lack of stability is detected as the DNA passes through this part of the polymerase. The incorrect base is excised and can be replaced with the correct one, as shown in figure 616. If both the parent and daughter strands are simple DNA, how does the enzyme discriminate which is the template strand and which is the incorrectly paired daughter strand? It looks at the level of methylation. The template strand has existed in the cell for a longer period of time, and therefore is more heavily methylated. Methylation also plays a role in the transcriptional activity of DNA, as described in Chapter 7 of Biochemistry. This system is very efficient, correcting most of the errors put into the sequence during replication. DNA ligase, which closes the gap between Osaki fragments, lacks proofreading ability. Thus, the likelihood of mutations in the lagging strand is considerably higher than the leading strand. DNA ligase seals the end as well as gaps. But lacks proofreading. Mismatch repair. Cells also have machinery in the G2 phase of the cell cycle for mismatch repair. These enzymes are encoded by genes MSH2 and MLH1, which detect and remove errors introduced in replication that were missed during the S phase of the cell cycle. These enzymes are homologous of mut-S and mut-L in prokaryotes, which serve a similar function. Mismatch repair seems to be a function in G2. MSH2 and MLH1. Nucleotide and base excision repair. Most of the repair mechanisms involve proteins that recognize damage or a lesion, remove the damage, and then use the complementary strand as a template to fill in the gap. Our cell machinery recognizes 2 specific types of DNA damage in the G1 and G2 cell cycle phases and fixes them through nucleotide excision repair or base excision repair. Nucleotide or just the base. of the nucleotide. Nucleotide. Excision. Repair. Ultraviolet light induces the formation of dimers between adjacent thymine residues in DNA. The formation of thymine dimers interferes with DNA replication and normal gene expression and distorts the shape of the double helix. Thymine dimers are eliminated from DNA by a nucleotide excision repair (NER) mechanism, which is a cut and patch process as shown in Figure 617. First, specific proteins scan the DNA molecule and recognize a lesion because of a bulge in the strand. An excision endonuclease then makes nicks in the phosphodiester backbone of the damaged strand on both sides of the thymine dimer and removes the defective oligonucleotide. DNA polymerase can then fill in the gap by synthesizing DNA in the 5' to 3' direction using the undamaged strand as a template. Finally, the nick in the strand is sealed by DNA ligase. So, TT dimers make a bulge in the DNA. Let's look at the example of that. Make a bulge. It says first specific proteins, they don't name them. An excision endonuclease breaks phosphodiester bonds. And then DNA polymerase can fill in the gap. And then DNA ligase, I imagine, recreates those phosphodiester bonds. One thing I wanted to add was about proofreading. Methylation is something that's kind of important. Mismatch repair. Let's just write down that in proofreading, I did write proofreading somewhere. Where did that go? Lacks proofreading, DNA ligase. In proofreading, methylation indicates original or older strand. So methylation is just present on older DNA. It also plays a role in transcriptional activity. So DNA has to be methylated to be transcribed. All right. Base excision repair. Alterations to bases can occur with other cellular insults. For example, thermal energy can be absorbed by DNA and may lead to cytosine deamination. This is a loss of an amino group from cytosine and results in the conversion of cytosine to uracil. Uracil should not be found in a DNA molecule and is thus easily detected as an error. However, detection systems exist for small non-helix distorting mutations in other bases as well. These are repaired by base excision repair. First, the affected base is recognized and removed by a glycosylase enzyme, leaving behind an apurinic, apyrimidinic AP site, also called an abasic site. The AP site is recognized by an AP endonuclease that removes the damaged sequence from the DNA. DNA polymerase and DNA ligase can then fill in the gap and seal the strand as described above. Base excision. With some heat can turn to uracil. And then glycosylase will remove, leading to a apurinic, apyrimidinic AP site. So a basic site. No base. And then AP nuclease. Endonuclease. Replaces that sugar, I guess, or just removes the sugar. And then DNA polymerase. And DNA ligase do their thing. All right. What is the difference between an oncogene and a tumor suppressor gene? The oncogene is the gene that causes a cancer cell. The tumor suppressor gene is a gene that makes tumor suppressing enzymes or proteins. So, right. 2. How does DNA polymerase recognize which strand is the template strand once the daughter strand is synthesized? Because the level of methylation of the parent strand will be greater than for the daughter strand. For each of the repair mechanisms below, in which phase of the cell cycle does the repair mechanism function? What are the key enzymes or genes specifically associated with each mechanism? That's a good point. Mismatch repair was G2. Nucleotide and base excision repair is both G1 and G2. So DNA polymerase is in S, G1, and G2. Key enzymes. Key enzymes or genes? I guess they need primers. Mismatch repair. Was in G2 phase. Key enzymes, MSH2, MLH1. Nucleotide excision repair is in G1 and G2. Base excision is also in G1 and G2. In nucleotide, you have excision endonuclease. In the base excision, you have glycosylase, followed by AP endonuclease. What are the key structural differences in the types of lesions corrected by nucleotide excision repair versus those corrected by base excision repair? Key structural differences is a dimer between T and T in the case of nucleotide excision repair, which causes a bulge. In base excision, you have cytosine turning into uracil, and that is RNA, not DNA. It doesn't bind in a double helix as well as RNA, or as DNA does. And specifically, deamination of cytosine. So let's look at cytosine real quick. Conversion of cytosine to uracil. So this nitrogen on top is lost or replaced by water. Cytosine to uracil on top there. Great. Let's check the answers real quick. Oncogenes, or more properly, proto-oncogenes, code for cell cycle promoting proteins. When mutated, the proto-oncogene becomes an oncogene, promoting rapid cell cycling. Tumor suppressor genes code for repair or cell cycle inhibiting proteins. When mutated, the cell cycle is allowed to proceed unchecked. The parent strand is more heavily methylated, whereas the daughter strand is barely methylated at all. This allows DNA polymerase to distinguish between the two strands. Number 3, we have polymerase in S. Isn't it in G1 and G2? DNA polymerase. And it has itself as the key enzyme. Great. Mismatch repair, G2, MSH2, MLH1, and it has the prokaryote versions too. Nucleotide excision repair, G1 and G2. Base excision, also G1 and G2. In nucleotide excision repair, you have your excision endonuclease, and in base excision repair, you have glycosylase and AP endonuclease. Number 4, nucleotide excision repair corrects lesions that are large enough to distort the double helix. Base excision repair corrects lesions that are small enough not to distort the double helix. They're saying that cytosine uracil doesn't distort the double helix. There you have it. I have to get some water, so I might as well take a break. We have one more section and then some questions. All right. Recombinant DNA and biotechnology. Now that we've reviewed the basics of DNA structure and function, we can discuss how this knowledge has been harnessed for a variety of research and treatment innovations. Recombinant DNA technology allows a DNA fragment from any source to be multiplied by either gene cloning or polymerase chain reaction (PCR). This provides a means of analyzing and altering genes and proteins. It also provides the reagents necessary for genetic testing, such as carrier detection, detecting heterozygote status for a particular disease, and prenatal diagnosis of genetic diseases. It is also useful for gene therapy. Additionally, this technology can provide a source of a specific protein, such as recombinant human insulin, in almost unlimited quantities. The process of creating recombinant DNA by gene cloning, and its benefits are shown in Figure 6.18. Recombinant DNA. PCR is your polymerase chain reaction. DNA cloning and restriction enzymes. DNA cloning is a technique that can produce large amounts of a desired sequence. Often the DNA to be cloned is present in a small quantity and is part of a heterogeneous mixture containing other DNA sequences. The goal is to produce a large quantity of homogeneous DNA for other applications. Cloning requires that the investigator ligate the DNA of interest into a piece of nucleic acid referred to as a vector, forming a recombinant vector. Vectors are usually bacterial or viral plasmids that can be transferred to a host bacterium after insertion of the DNA of interest. The bacteria are then grown in colonies, and a colony containing the recombinant vector is isolated. This can be accomplished by ensuring that the recombinant vector also includes a gene for antibiotic resistance. Antibiotics can then kill off all of the colonies that do not contain the recombinant vector. The resulting colony can then be grown in large quantities. Depending on the investigator's goal, the bacteria can then be made to express the gene of interest, generating large quantities of recombinant protein, or can be lysed to re-isolate the replicated recombinant vectors, which can then be processed by restriction enzymes to release the cloned DNA from the vector. So cloning requires a vector, bacterial or viral, plasmids specifically. Restriction enzymes. Restriction endonucleases are enzymes that recognize specific double-stranded DNA sequences. These sequences are palindromic, meaning that the 5-3 sequence of one strand is identical to the 5-3 sequence of the other strand in antiparallel orientation. Restriction enzymes are isolated from bacteria, which are their natural source. In bacteria, they act as part of a restriction and modification system that protects the bacteria from infection by DNA viruses. Once a specific sequence has been identified, the restriction enzyme can cut through the backbone of the double helix. Thousands of restriction enzymes have been studied, and many are commercially available to laboratories, allowing us to process DNA in very specific ways. Some restriction enzymes produce offset cuts, yielding sticky ends on the fragments as shown in Figure 6.19. Sticky ends are advantageous in facilitating the recombination of a restriction fragment with the vector DNA. The vector of choice can also be cut with the same restriction enzyme, allowing the fragments to be inserted directly into the vector. Restriction endonuclease. Palindromic. Oops. Sticky ends. DNA vectors contain at least one sequence, if not many, recognized by restriction enzymes. A vector also requires an origin of replication and at least one gene for antibiotic resistance to allow for selection of colonies with recombinant plasmids as described above. The formation of a recombinant plasmid is shown in figure 620. So it won't be replicated if you don't have an origin of replication. I guess you want to replicate it to clone these, to produce these. And you need antibiotic resistance to isolate them from their cousins that don't have the plasmids that you want, or that don't have the DNA. What do you call what you add? The fragment, the DNA fragment? For selection, this also needs antibiotic resistance for selection. All right. DNA libraries and cDNA. DNA cloning can be used to produce DNA libraries. DNA libraries are large collections of known DNA sequences. In sum, these sequences could equate to the genome of an organism. To make a DNA library, DNA fragments often digested randomly are cloned into vectors and can be utilized for further study. Libraries can consist of either genomic DNA or cDNA. Genomic libraries contain large fragments of DNA and include both coding, exon, and non-coding intron regions of the genome. cDNA, or complementary DNA, libraries are constructed by reverse transcribing process mRNA, as shown in figure 621. As such, cDNA lacks non-coding regions, such as introns, and only includes the genes that are expressed in the tissue from which the mRNA was isolated. For that reason, these libraries are sometimes called expression libraries, while genomic libraries contain the entire genome of an organism. Genes may by chance be split into multiple vectors. Therefore, only cDNA libraries can be used to reliably sequence specific genes and identify disease-causing mutations, produce recombinant proteins such as insulin clotting factor or vaccines, or produce transgenic animals. Several of these applications are discussed in more detail in subsequent sections of this chapter. Table 6.3 contrasts some of the characteristics of genomic and cDNA libraries. So, remember that exons are coding regions, intron is non-coding, and cDNA is made by reverse transcribing RNA. So they lack exons. It only represents the introns. And we have a chart of differences between genomic libraries and cDNA, or expression libraries. Enzymes to make the library, nucleases and ligases, while you use transcriptases in cDNA because you need to reverse transcribe. Genomic libraries have non-expressed sequences. cDNA does not. Cloned genes are completed sequences, not necessarily in genomic libraries, but they are in cDNA. Cloned genes contain introns. I guess that's just because genomic libraries have a large number of DNA, but you can't be sure that you get them all. While cDNA, if it's transcribed and it's a functional like protein, then at least that little bit is fully functional. Cloned genes contain introns. Yes, in genomic libraries, not in cDNA. Promoter and enhancer sequences present. Yes, in the genomic, but not in the cDNA. So it has to do with gene expression. Genes can be expressed in cloning host. Recombinant proteins, not in the genomic, yes, in the cDNA. Why can't the genes be expressed in the cloning host, recombinant proteins? I guess you don't put recombinant proteins in the genomic library because then you'd corrupt the library. Well, as cDNA, I guess you would have recombinant proteins because it's a library of expression. Finally, can be used for gene therapy or constructing transgenic animals. Expression libraries can, genomic libraries can't. Hybridization. Another tool often used by researchers is called hybridization. Hybridization is the joining of complementary base pair sequences. This can be DNA recognition or DNA-RNA recognition. This technique uses two single-stranded sequences and is a vital part of polymerase chain reaction and southern blotting. Polymerase chain reaction. PCR is an automated process that can produce millions of copies of a DNA sequence without amplifying the DNA in bacteria. PCR is used to identify criminal suspects, familiar relationships, and disease-causing bacteria and viruses. Knowing the sequence that flank the desired region of DNA allows for the amplification of the sequence in between. A PCR reaction requires primers that are complementary to the DNA that flanks the region of interest. Nucleotides, dATP, dTTP, dCTP, and dGTP, and DNA polymerase. The primer has high GC content, 40 to 60% is optimal, as the additional hydrogen bonds between G and C confer stability. The reaction also needs heat to cause the DNA double helix to melt apart. Unfortunately, the DNA polymerase found in the human body does not work at high temperatures. Thus, the DNA polymerase from Thermus aquaticus, a bacteria that thrives in the hot springs of Yellowstone National Park at 70 degrees Celsius, is used instead. During PCR, the DNA of interest is denatured, replicated, and then cooled to allow reannealing of the daughter strands with the parent strands. This process is repeated several times, doubling the amount of DNA with each cycle, until enough copies of the DNA sequence are available for further testing. So it's a way of rapidly multiplying DNA. PCR. Should I write hybridization? Both so hybridization is used in both PCR and southern blotting. So we're just joining DNA to its complement. PCR uses primers that recognize the sequence. And he uses DNA polymerase from Thermus aquaticus. Southern blotting. Gel electrophoresis is a technique used to separate macromolecules such as DNA and proteins by size and charge. Electrophoresis of proteins was discussed in detail in Chapter 3, but DNA can be separated in a similar way. All molecules of DNA are negatively charged because of the phosphate groups in the backbone of the molecule. So all DNA strands will migrate toward the anode of an electrochemical cell. The preferred gel for DNA electrophoresis is agarose gel. And just like proteins in polyacrylamide gel, the longer the DNA strand, the slower it will migrate in the gel. Agarose instead of polyacrylamide. DNA is negatively charged, as we mentioned earlier. Gel electrophoresis is often used while performing a southern blot. A southern blot is used to detect the presence and quantity of various DNA strands in a sample. DNA is cut by restriction enzymes and then separated by gel electrophoresis. The DNA fragments are then carefully transferred to a membrane, retaining their separation. The membrane is then probed with many copies of a single-stranded DNA sequence. The probe will bind to its complementary sequence and form double-stranded DNA. Probes are labeled with radioisotopes or indicator proteins, both of which can be used to indicate the presence of a desired sequence. So southern blotting is the use of phoresis along with a radioisotope labeled probe. DNA sequencing. DNA sequencing has revolutionized the world that we live in. The applications of this technique are far-reaching, from the medical field to criminal courts. A basic sequencing reaction contains the main players from replication, including template DNA primers, an appropriate DNA polymerase, and all 4 deoxyribonucleotide triphosphates. In addition, a modified base called dideoxyribonucleotide is added in lower concentrations. Dideoxyribonucleotides, DDATP, DDCTP, DDGTP, and DDTTP, contain a hydrogen at C3 rather than a hydroxyl group. Thus, one of these modified bases has been incorporated. Once one of these modified bases has been incorporated, the polymerase can no longer add to the chain. Eventually, the sample will contain many fragments, as many as the number of nucleotides in the desired sequence, each one of which terminates with one of the modified bases. These fragments are then separated by size using gel electrophoresis. The last base for each fragment can be read, and because gel electrophoresis separates the strands by size, the bases can easily be read in order. DNA sequencing. Let's look at that on the 3 carbon of the sugar. Because this oxygen is also removed, the 3' end of the sugar cannot be used to make further bonds. And so that ends the DNA sequence right there, making these fragments that you then analyze. C3'. Applications of DNA technology. In addition to its utility as a research tool, DNA biotechnology has led to a number of therapeutic breakthroughs, ranging from gene therapy, described in this section, to development of personalized chemotherapeutic regimes in cancer by genotyping the tumor cells. DNA technology is also used in industry, including the development of genetically modified foods that are enriched with specific nutrients and testing of the environment for risk assessment and cleanup procedures. As mentioned previously, DNA technology also plays a key role in forensic pathology and crime scene investigation. This is likely only the beginning, as biotechnology continues to be an active area of research. Gene therapy. Gene therapy now offers potential cures for individuals with inherent diseases. Gene therapy is intended for diseases in which a given gene is mutated or inactivated, inactive, giving rise to pathology. By transferring a normal copy of the gene into the affected tissue, the pathology should be fixed, essentially curing the individual. For instance, about half of children with severe combined immunodeficiency, SCID, have a mutation in the gene encoding the gamma chain common to several of the interleukin receptors. By placing a working copy of the gene for the gamma chain into a virus, one can transmit the functional gene into human cells. The first successful case of gene therapy was for SCID caused by a different mutation in 1990. For gene replacement therapy to be a realistic possibility, efficient gene delivery vectors must be used to transfer the cloned gene into target cells' DNA. Because viruses naturally infect cells to insert their own genetic material, most gene delivery vectors in use are modified viruses. A portion of the viral genome is replaced with the cloned gene such that the virus can infect but not complete its replication cycle, as shown in Figure 6.22. Randomly integrated DNA poses a risk of integrating near and activating a host oncogene. Among the children treated for SCID, a small number have developed leukemias, cancers of white blood cells. Retroviral gene therapy. Transgenic and knockout mice. Once DNA has been isolated, it can be introduced into eukaryotic cells. Actually, applications of DNA technology. Let's write something down. Applications, gene therapy, and in gene therapy, small number of patients develop cancer. That's just the risk. Transgenic and knockout mice. Once DNA has been isolated, it can be introduced into eukaryotic cells. Transgenic mice are altered at their germ line by introducing a cloned gene into fertilized ova or into embryonic stem cells. The cloned gene that is introduced is referred to as a transgene. If the transgene is a disease-producing allele, the transgenic mice can be used to study the disease process from early embryonic development through adulthood. A similar approach can be used to produce knockout mice, in which a gene has been intentionally deleted, knocked out. These mice provide valuable models in which to study human diseases. Transgenetic and knockout mice. There are different approaches to developing transgenic mice. A cloned gene may be micro-injected into the nucleus of a newly fertilized ovum. Rarely, the gene may subsequently incorporate into the nuclear DNA of the zygote. The ovum is implanted into a surrogate mother, and if successful, the resulting offspring will contain the transgene in all of their cells, including their germ-line cells, or gametes. Consequently, the transgene will also be passed to their offspring. The transgene coexists in the animal with their own copies of the gene, which have not been deleted. This approach is useful for studying dominant gene effects, but is less useful as a model for recessive disease because the number of copies of the gene that insert into the genome cannot be controlled. The transgenic mice may each contain a different number of copies of the transgene. This method is demonstrated in Figure 6.23. So the mother doesn't have it, the kids have it, but they all have different copies, different number of copies. Embryonic stem cell lines can also be used for developing transgenic mice. Advantages of using stem cell lines are that cloned genes can be introduced in cultures, and that one can select for cells with the transgene successfully inserted. So let's write down germline versus embryonic stem cell lines. Embryonic stem cell lines can also be used for developing transgenic mice. Advantages of using stem cell lines are that the cloned genes can be introduced in cultures and that one can select for cells with the transgene successfully inserted. The altered stem cells are injected into developing blastocysts and implanted into surrogate mothers. The blastocyst itself is thus composed of two types of stem cells, the ones containing the transgene and the original blastocyst cells that lack the transgene. The resulting offspring is a chimera, meaning that it has patches of cells, including germ cells, derived from each of the two lineages. This is evident if the two cell lineages, transgenic cells and host blastocysts, come from mice with different coat colors. The chimera will have patchy coats of two colors, allowing them to be easily identified. These chimera can then be bred to produce mice that are heterozygous for the transgene and mice that are homozygous for the transgene. All right. So embryonic stem cells, chimera, and separate heterozygous homozygous populations. Separate populations of homozygous and heterozygous mice. The first one, you just had them mixed up. You can't be sure if what you have is heterozygous or homozygous. This is a system in which you can identify, like easily isolate, using chimera. Interesting. Safety and ethics. The different procedures and techniques that have been reviewed provide great insight for researchers in many different fields of study. However, it is also important to acknowledge the potential risks associated with these technologies. Safety concerns such as increased resistance in viruses and bacteria can impact both humans and the environment in which we live. Ethical dilemmas arise. Is it ethical to test for life-threatening genetic diseases and potentially terminate a pregnancy based on the results? What about testing for eye or hair color? What are the ethical questions around choosing human test subjects? If a disease-causing gene were found in one individual of a family, does this need to be communicated to other relatives at risk, potentially violating principles of privacy? Is it permissible to carry out potentially risky therapy in an individual whose illness makes him or her unable to communicate? The medical communities and bioethicists at large continue to wrestle with this question. How much should we meddle with our own genetic makeup? Great. Some ethics there. When creating a DNA library, what are some of the advantages of genomic libraries? What about cDNA libraries? Advantages of genomic libraries is you have all the DNA. Just about. cDNA, I guess you have sequences that are relevant because you have like sequences that code for specific proteins. In genomic, you have fragments of everything. I'm sure it takes like a bunch of analysis to get the actual sequence, but I think we could do that now as well. What does PCR accomplish for a researcher? What about southern blotting? PCR will greatly amplify a sequence. I guess southern blotting is to identify. During DNA sequencing, why does DNA polymer stop growing once a dideoxy ribonucleotide is added? Because the 3 carbon on the ribose is deoxygenated. It can't be used in the polymer backbone. And so the DNA sequence ends right there. What is the difference between a transgenic and a knockout mouse? Transgenic has added DNA. Knockout mouse has like knocked out or removed DNA. Let's check the answers for those. Number 1. Genomic libraries include all the DNA in an organism's genome, including non-coding regions. This may be useful for studying DNA in introns, centromeres, or telomeres. cDNA libraries only include express genes from a given tissue, but can be used to express recombinant proteins or to perform gene therapy. 2. PCR increases the number of copies of a given DNA sequence, can be used for a sample containing very few copies of the DNA sequence. Southern blotting is useful when searching for a particular DNA sequence because it separates DNA fragments by length and then probes for a sequence of interest. Number 3, dideoxyribonucleotides lack the 3' hydroxyl group that is required for DNA strand elongation. Thus, once a dideoxyribonucleotide is added to a growing DNA molecule, no more nucleotides can be added because dideoxyribonucleotides have no 3' hydroxyl group with which to form a bond. Number 4, transgenic mice have a gene introduced into their germline or embryonic stem cells to look at the effects of that gene. They are therefore best suited for studying the effects of dominant alleles. Knockout mice are those in which a gene of interest has been removed rather than added. Transgenic versus knockout. Better for dominant. All right. Some practice questions. In a single strand of a nucleic acid, nucleotides are linked by A. Hydrogen bonds, B. Phosphodiester bonds, C. Ionic bonds, D. Van der Waals forces. Number 1. Nucleic acid nucleotides are linked by phosphodiester bonds, B. Are they ionic bonds? No, they're covalent bonds. 2. Which of the following statements regarding differences between DNA and RNA is false? In cells, DNA is double-stranded, whereas RNA is single-stranded. Sounds good. DNA uses the nitrogenous base thymine. RNA uses uracil. Sounds right. The sugar in DNA is deoxyribose. The sugar in RNA is ribose. That is also true, right? DNA strands replicate in a 5-3 direction, whereas RNA is synthesized in the 3-5 direction. That's false. It's still synthesized in the 5-3 direction, I imagine. I don't know. 3. Which of the following DNA sequences would have the highest melting temperature? So a lot of CG makes it higher melting point. So AT, it's the G's and C's we want. So let's just count them, I guess. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4. 1, 2, 3, and 1, 2, 3. So A is the clear winner, the most C's and G's. 4. Which of the following biomolecules is least likely to contain an aromatic ring? Proteins, purines, carbohydrates, or pyrimidines? I guess carbohydrates. Proteins, purines, and pyrimidines all have aromatic rings, right? These are not aromatic rings. Pyrimidines do. Purines don't. So I forgot to label these purines and pyrimidines. Never mind, pyrimidines don't have aromatic rings. Proteins do, purines do. Carbohydrates, do they have aromatic rings? I guess they could. What's a carbohydrate? Carbohydrates meaning sugars. When ribose makes a ring, it's not conjugated. It's not a aromatic. Well, it's not aromatic. It still has pi bond character. We're going to call it aromatic. We're going to say the sugar is not aromatic. We're going to go with carbohydrates. C. 5. For a compound to be aromatic, all of the following must be true, except the molecule is cyclic, the molecule contains 4n plus 2 pi electrons, the molecule contains alternating single and double bonds, the molecule is planar. They all seem true to me. Four n plus 2 electrons. That's the pi electrons. That's Huckel's rule. Compound is cyclic, compound is planar, compound is conjugated, and the compound has 4n plus 2. Four n plus 2. So conjugated could be either alternating single or double bonds or lone pairs. So I guess since this leaves out lone pairs, it's the least accurate. It doesn't have to be double bonds, like in the case of nitrogen, which adds a lone pair rather than a double bond. So we'll say C is the one that's not necessary. 6. Which of the following enzymes is not involved in DNA replication? Primase, DNA ligase, DNA polymerase, or telomerase? Telomerase makes telomeres. I'm going to say that's not a part of replication because all the others are. Primase, polymerase, ligase. Telomerase was separate. Seven, how is cDNA best characterized? cDNA results from a DNA transcript with non-coding regions removed. Now, cDNA results from the reverse transcription of processed mRNA. That sounds good. cDNA is the abbreviation for deoxycytosine. No. D, cDNA is the circular DNA molecule that forms the bacterial genome. No. B, reverse transcription of processed mRNA. 8. Which of the following statements regarding polymerase chain reactions is false? Human DNA polymerase is used because it is the most accurate. Well, that's not true. B. A primer must be prepared with complementary sequence to part of the DNA of interest. Sure. Repeated heating and cooling cycles allow the enzyme to act specifically and replace helicase. Sure. D. Each cycle of the polymerase chain reaction doubles the amount of DNA of interest. Also sounds plausible. A is the one that is not true. DNA polymerase degrades, and so you use that that Yellowstone polymerase instead. 9. Restriction endonucleases are used for which of the following? Gene therapy, southern blotting, DNA repair. Restriction endonuclease. It's used for gene therapy, southern blotting, DNA repair. Not like yeah, I guess you use them for all of them, right? Southern blotting, does southern blotting use restriction endonucleases? Sure, why not? Does DNA repair use restriction endonucleases? Excision endonuclease is repair. And for base excision, there's an AP endonuclease. It's used probably in PCR and using cloning. It's probably used in cDNA. Maybe not. So recombinant DNA for sure. So gene therapy. Southern blotting. Restriction enzymes. So restriction endonucleases, sure. Gene therapy, southern blotting, DNA repair. Does the natural process of DNA repair use restriction enzymes? No. Excision and restriction, or excision and nucleotide excision endonuclease. And polymerase really does the proofreading, huh? DNA repair, I imagine. Polymerase does the... How does repair happen? We're going to go with 1 and 2. No, there's no 1 and 2 option. So 1, 2, and 3. Right, D. Great. 10. How does prokaryotic DNA differ from eukaryotic DNA? Prokaryotic DNA lacks nucleosomes. Eukaryotic DNA has telomeres. Prokaryotic DNA is replicated by a different DNA polymerase. Eukaryotic DNA is circular and not restricted by centromeres. So the only one that's false is 4. 1, 2, and 3 all sound pretty good. Prokaryotic lacks nucleosomes. There's just in one large plasmid, circular plasmid. Eukaryotic DNA has telomeres, that's true. Since it's linear, it needs telomeres to hold its ends knotted. Since prokaryotic DNA is circular, it doesn't have to have like the ends tied off. DNA polymerase in prokaryotes are like the 1, 2, and 3 rather than the alpha, beta, gamma, delta, epsilon. Prokaryotic DNA is not circular. That's not true. So 10D11. Why might uracil be excluded from DNA but not RNA? Uracil is much more difficult to synthesize. Uracil binds adenine too strongly for replication. Cytosine degradation results in uracil. Uracil is used as a DNA synthesis activator. I bet it has to do with the binding. It binds to adenine too strongly. Let's go with B. 12. Tumor suppressor genes are most likely to result in cancer through loss of function mutations, gain of function mutations, overexpression, or proto-oncogene formation. Tumor suppressor genes, loss of function would cause cancer through that mechanism, right? 13. Which of the following is an ethical concern of gene sequencing? Gene sequencing is invasive, thus the potential health risk must be thoroughly explained. Gene sequencing impacts relatives, thus privacy concerns may be raised. Gene sequencing is very inaccurate, which increases anxiety related to findings. Or D, gene sequencing can provide false negative results, giving a false sense of security. They talked about the impact on privacy. So let's go with the impact on privacy. 14. Which of the following is not a difference between heterochromatin and euchromatin? Euchromatin has areas that can be transcribed, whereas heterochromatin is silent. That's true. Heterochromatin is tightly packed, whereas euchromatin is less dense. That's true. Heterochromatin stains darkly, whereas euchromatin stains lightly. That's also true. Heterochromatin is found in the nucleus, whereas euchromatin is found in the cytoplasm. That's the only one that is not the difference. They're both in the nucleus. 15. During which phase of the cell cycle are DNA repair mechanisms least active? G1, S, G2, M. I want to say S. Maybe it's M. What are the cell cycle phases again? Cell cycle. Phases of the cell cycle. G1 is interphase. So G1, S, and G2. M is mitosis. So DNA synthesis is interphase S. Cell growth is G1 and G2. But in mitosis, I think it's not doing DNA repair. At that point, it's just splitting. I'm going to go with D. During the mitotic M phase, the cell divides its copied DNA in cytoplasm to make two new cells. So all the replication and stuff is happening in the other cycles. All right, let's see how we did. Number 1, B. 2, D. 3, A. 4, C. 5, C. 6 is going to be C, not D. During DNA replication, the strands are separated by DNA helicase. At the replication fork, primase, choice A, creates a primer for the initiation of replication, which is followed by DNA polymerase. On the lagging strand, Ozaki fragments form and are joined by DNA ligase, choice B. After the chromosome has been processed, the ends called telomeres are replicated with the assistance of the enzyme telomerase. Choice D. RNA polymerase, distinct from primase, is responsible for transcription. RNA polymerase is responsible... Oh, not DNA polymerase, but RNA polymerase. That should be a giveaway. I kind of assumed it said DNA polymerase. But yes, RNA polymerase is not even in DNA replication. Telomerase is. You still need to make telomeres to seal off the ends. Jeez. 7 is going to be B. 8 is going to be A. 9 is going to be D. 10 is D. 11 is C, not B. One common DNA mutation is the transition from cytosine to uracil in the presence of heat. DNA repair enzymes recognize uracil and correct this error by excising the base and inserting cytosine. RNA exists only transiently in the cell, such that cytosine degradation is insignificant. Where uracil is used in DNA under normal circumstances, it would be impossible to tell if a base should be uracil or if it is a damaged cytosine nucleotide. Cytosine degradation results in uracil. So if you had a uracil, you wouldn't know that it wasn't a cytosine. But in RNA, it doesn't matter because it's not going to stick around that long. Well, there you have it. 12 is A, 13 is B, 14 is D, and 15 is D. That is it for DNA and biotechnology. Next time, Chapter 7, RNA and the genetic code.